Welcome back to this next video in which we are discussing multiple sclerosis. So we have now finished discussing the pathogenesis of multiple sclerosis, and in this next video we're going to move on to now discussing the current drug treatment for multiple sclerosis. However, before we do that, allow me to summarize the story of the pathogenesis of multiple sclerosis now that we have the entire story. So we have seen that in order for an individual to develop either multiple sclerosis or a clinically isolated syndrome, there are three major stages in the pathogenesis that they have to go through. Stage 1 is that they must develop an adaptive immune system that is capable of attacking a protein of the central nervous system myelin. And because we don't know what protein specifically it is within the central nervous system myelin that gets attacked, we've agreed to just call that protein the central nervous system myelin protein. So that's stage 1. Stage 2 is that autoreactive adaptive immune system response needs to actually be activated, and we've discussed the fact that we believe that occurs when the individual gets infected in some other tissue by a pathogen that has an antigen that is capable of activating the autoreactive adaptive immune response. So that's stage 2. So by the end of stage 2, you now have effector CD4 positive T cells that are directed against a peptide fragment of the CNS myelin protein, and antibodies that are directed against an epitope of the central nervous system myelin protein present within the blood. So this part of the picture is where you've got to by the end of stage 2. However, if you just get up to the end of stage 2 and then don't go through stage 3, that will not cause any disease. Them being in the blood is not a problem. They only trigger the formation of white matter lesions if they actually get into the brain tissue. So stage 3 is extremely important, and stage 3 is blood-brain barrier penetration. So what has to happen then in stage 3 is the individual has to suffer from an episode where blood-brain barrier penetration occurs in certain areas of the central nervous system. So the blood-brain barrier has to, for some reason, allow the antibody molecules and the effector helper T cells to come from the bloodstream into the brain tissue in regions within the central nervous system, which we've marked in green here on our picture. And in those regions where you get the blood-brain barrier penetration, that's where you will then get white matter lesions forming. So without an episode of blood-brain barrier penetration in regions of the central nervous system, you are not going to develop any white matter lesions, and therefore you are not going to develop any clinical disease. Now, we discussed at the end of the previous video possible explanations as to what causes an episode of blood-brain barrier penetration. And one of the possible explanations is that it is high circulating levels of pro-inflammatory mediators. So the two most important pro-inflammatory mediators are interleukin-1 and tumor necrosis factor alpha, beautifully written here. Now, when you are very, very ill, when you have an infection, potentially flu, let's say, you will get inflammation occurring in the local tissue that is infected. If there is a lot of tissue infected, then you'll get a lot of inflammation. And when you have inflammation occurring in a portion of tissue, the inflammatory mediators released there don't just stay in that local area, they go into the bloodstream as well. So our two most important examples, interleukin-1 and TNF-alpha, they end up going into the bloodstream. And the presence of high circulating levels of pro-inflammatory mediators is what causes the systemic symptoms of illness. However, it's also plausible that it might be the high circulating levels of pro-inflammatory mediators in a severe infection that are going to lead to an episode of blood-brain barrier penetration, because think about this. What do interleukin-1 and tumor necrosis factor alpha do? They work on receptors on endothelial cells to change the behavior of those endothelial cells. They tell endothelial cells to start allowing proteins from the bloodstream to cross into the tissue fluid and also to start recruiting white blood cells into the tissue fluid from the bloodstream. So it is plausible then that if you have very high levels of interleukin-1 and tumour necrosis factor alpha circulating within the bloodstream, that this could affect the capillaries in these regions of the central nervous system and then cause them to allow antibody molecules, some antibody molecules and some effector helper T cells across from the bloodstream into the central nervous system tissue, and that will kickstart the formation of a white matter lesion. And this is consistent with the fact that many people with multiple sclerosis say that they get episodes of new white matter lesion formation when they are ill in some other way. So for instance, when they've got a horrible episode of flu, or when they've got a horrible episode of gastroenteritis, i.e. when their levels of circulating inflammatory mediators will be very high. So this is potentially the explanation as to what causes these episodes of blood-brain barrier penetration. But be aware that this is a very, very crucial stage in the pathophysiology. If you never get stage 3 occurring, then you will not get any disease, even if you've got stage 1 and stage 2 having occurred. Now, another thing to point out is that if we believe this theory of what causes this episode of blood-brain barrier penetration, i.e. that it's high circulating mediators, circulating pro-inflammatory mediators specifically, then it may be the case that stage 3 and stage 2 often occur together, because remember, stage 2 was caused by a pathogen infecting some tissue within the body and then managing to activate the autoreactive adaptive immune system response. That pathogen might also be capable, therefore, of actually causing the first episode of blood-brain barrier penetration, because it might lead to very high levels of circulating pro-inflammatory mediators. So be aware that stage 2 and stage 3 might actually occur at the same time, and therefore you might get your first episode of white matter lesion formation exactly when you get the activation of the autoreactive adaptive immune system response. Okay, right. So once you've got blood-brain barrier penetration occurring in these regions within the central nervous system, of course what will happen is the antibody molecules and the effector helper T cells will come into the brain tissue in those regions, and they will result in the formation of a white matter lesion, as indeed this picture shows here. And in that white matter lesion, you're going to get demyelination of the axons present there. So the myelin sheaths are going to be lost. But remember, the axons themselves are usually not destroyed, and the oligodendrocyte cell bodies are often not destroyed either. So this means that potentially the white matter lesion can heal itself. Indeed, what will happen is after all the myelin sheaths have been destroyed in a white matter lesion, then the inflammation will go down. So the inflammatory cells will gradually leave that area and a bit of normality will return. After the inflammation has stopped in the white matter lesion, it is then possible that the oligodendrocytes may remyelinate the axons and you may get healing of that white matter lesion. That doesn't occur always, but it can occur. But be aware that the inflammation in a white matter lesion will not go on forever. It will go on until the myelin sheaths have been destroyed. And it's important to understand that to later justify why we give certain drugs acutely in an episode of white matter lesion formation in multiple sclerosis. But more about that in a moment. Finally, just to say a little bit about the difference between the different forms of MS and a clinically isolated syndrome. So in someone who just suffers from a clinically isolated syndrome, they are only going to get one episode of stage 3 occurring. They're only going to get one episode of white matter lesion formation, and then for the rest of their life they're going to be very lucky. They're not going to get another episode of blood-brain barrier penetration, and therefore they're not going to get another episode of white matter lesion formation. Coming on to the different forms of MS, of course in MS the definition is that you get recurrent episodes of white matter lesion formation, so you must therefore have recurrent episodes of blood-brain barrier penetration. About the different types of MS, of course 85% of people originally develop relapsing remitting MS. In this form of MS, in between episodes of new white matter lesion formation, the previous white matter lesions are capable of healing. So we've discussed the fact that after all the myelin sheaths have been destroyed, the inflammation will subside, and after that, either healing can occur or healing will not occur. In relapsing remitting multiple sclerosis, healing is going to occur quite well, so the oligodendrocytes are going to start remyelinating the axons, and therefore that area of tissue can regain its normal function and the symptoms will go away. In the progressive forms of multiple sclerosis, either primary progressive multiple sclerosis, which 15% of people originally suffer from, or secondary progressive multiple sclerosis, which unfortunately 2/3 of the people who start off with relapsing remitting multiple sclerosis will then develop, that's where the white matter lesions unfortunately do not properly heal afterwards. So the inflammation will still subside after all the myelin sheaths have been destroyed. The inflammation will subside, so the inflammatory cells will go away, but then you don't get the same sort of remyelination occurring, and therefore that tissue doesn't ever regain its function properly back again. So in primary progressive multiple sclerosis, you never have white matter lesions that properly heal, and therefore you just collect more and more neurological deficits. And in secondary progressive multiple sclerosis, you originally had relapsing remitting multiple sclerosis, and then somehow your body loses the ability to actually heal the white matter lesions properly, and you turn over to a progressive form of multiple sclerosis. Okay, and be aware also that at the same time we will have to have maintenance of the autoreactive adaptive immune response, and that will be done by when you have white matter lesions that are actually active, i.e. the inflammation is occurring, the antigen-presenting cells, so macrophages and microglial cells, will be draining in lymphatic vessels that do exist within the brain down to cervical lymph nodes and presenting the peptide fragment of the central nervous system myelin protein that we have autoreactive CD4 positive T cells directed against to memory CD4 positive T cells. In addition, you'll also be getting the central nervous system myelin protein being chucked out by the macrophages and the microglial cells into the tissue fluid to drain in the lymphatic vessels to then go and activate the memory B cells to maintain the autoimmune disease or the autoimmune adaptive immune response, so that you still maintain effector helper T cells and antibodies directed against the central nervous system myelin protein within the blood. Okay, so there's my summary of the pathogenesis. Let us now turn on to the drug treatment for multiple sclerosis. So I'll just put a little title here. So we're now going to discuss the treatment of multiple sclerosis, or the current treatment, I should say, of multiple sclerosis. So I'm going to split our discussion of treatment into two separate categories. There is what I will call the acute treatment of multiple sclerosis, and indeed also of clinically isolated syndromes, and then there is the preventative, or to give it an even fancier name, the prophylactic treatment. So let me just justify what these names actually mean. So the acute treatment for multiple sclerosis or a clinically isolated syndrome is the treatment that is given to someone who has just suffered from another episode, or their first episode maybe, of white matter lesion formation. So when someone has just acutely suffered from an episode of white matter lesion formation, of course they've just developed horrible neurological symptoms, what treatment do you give them to try and help them heal, to try and help the symptoms go away? That's the acute treatment. Whereas the prophylactic treatment is the treatment that you give to someone with multiple sclerosis rather than just a clinically isolated syndrome, to try and prevent them getting future episodes of white matter lesion formation. So this is how we actually treat an episode of white matter lesion formation that's actually occurred. This is how we try to prevent future white matter lesions from actually occurring. So let's start off with the acute treatment. So acute treatment you can give glucocorticoid drugs. Oral glucocorticoids can be given for the acute treatment of an episode of white matter lesion formation. So before I try and explain what these are actually going to do to help the person who's just been through an episode of white matter lesion formation, let me give you some examples of glucocorticoid drugs. So oral glucocorticoids, I'm going to give you 6 names here. So this is the listy bit. So a very important one is the drug prednisone. Now prednisone is actually a prodrug, so when you take it orally, it's converted in the liver to the active drug, and the active drug is called prednisolone. Now, you can actually give prednisolone straight away. So some people use prednisone, some people use prednisolone. Prednisolone and prednisone will do the exact same thing, it's just one is a prodrug, one is the actual active drug. So I'll say that again, prednisone is a prodrug, it will be converted in the liver into something called prednisolone, and prednisolone will actually circulate within the bloodstream and do what it's meant to do. You can, however, just give prednisolone straight away and then circumvent the need for the liver to convert prednisone into the active drug. There then is a modified version of prednisolone that is also effective at doing what prednisolone does, and this is called methylprednisolone. So that's another variant of prednisolone, does exactly the same thing as prednisolone. More examples of glucocorticoids. Hydrocortisone is a very important example, and this is actually just another name for the endogenous glucocorticoid called cortisol. 2 final examples. We have a drug called dexamethasone, a very powerful oral glucocorticoid. And then finally, a drug called triamcinolone. So triamcinolone. So that's the listy bit over. Those are 6 examples of oral glucocorticoids. So let me now try and explain what these are going to do. So glucocorticoids are very powerful anti-inflammatory drugs. We use them all over the place in medicine, very, very powerful drugs. And the way they work is by hijacking an endogenous system that your body has to reduce inflammation. So in your body, you have a hormone called cortisol. So I'll put this over here. So in your body, you have a hormone called cortisol that has a natural anti-inflammatory effect. It can reduce inflammation within the bloodstream. It's secreted by the adrenal glands, and it can go into the bloodstream and go into tissues and reduce inflammation in tissues. So the more cortisol you have, the more anti-inflammatory effect you'll be having all over the body. All of these drugs, well, not prednisone because it's a prodrug, but all of these other ones, these 5 ones here, they are going to hijack the same signaling pathway that cortisol uses to induce that anti-inflammatory effect. Indeed, hydrocortisone is the exact same molecule as cortisol. We use a different name for it when we're actually giving it exogenously, but it is the exact same molecule. So they're going to hijack a natural system. Now, you might leap to conclusions here. You might think, so we're giving an anti-inflammatory drug that will reduce down the inflammation in these white matter lesions, and therefore it will reduce the damage to the central nervous system. That's why we're going to do this. It's going to reduce the damage to the central nervous system that's happening in this episode of white matter lesion formation. And I'm afraid the answer is no. We don't think that giving glucocorticoids to someone who's just had an episode of white matter lesion formation actually reduces the overall damage that's going to occur. And the reason is we think we've given them too late. By the time that someone presents having had an episode of white matter lesion formation, the white matter lesions have formed. The white matter lesions have destroyed the myelin sheaths within the regions that they've affected. By the time they present with the neurological symptoms, the myelin, we believe, has almost all been destroyed in the white matter lesion. So actually, the point that the lesion is at is the myelin's been destroyed, and now it's getting ready to actually go into the healing phase. Now the inflammation is going to go, the inflammatory cells are going to leave that place, and then the healing process may well begin, i.e. you may get the remyelination occurring by the oligodendrocytes if we're talking about relapsing remitting multiple sclerosis. However, the point is that by the time they develop the neurological symptoms and present to hospitals, it's too late. The damage has been done. So giving the glucocorticoids isn't actually going to stop myelin destruction because the myelin destruction's actually happened already. So why do we give anti-inflammatory drugs at all then? Well, the idea is that these speed up the recovery. They speed up the removal of the inflammatory cells from the actual white matter lesions. So remember, the white matter lesion has formed, the myelin has now gone by the time that they've got the neurological symptoms, and what's now meant to follow is the immune cells are going to leave that area and some sort of normality is going to return to that portion of the central nervous system and potentially remyelination may occur. We believe that by giving people glucocorticoids, we speed up the removal of the inflammatory cells from the white matter lesions and therefore speed up recovery. So I want to stress that very much so here. We give glucocorticoids as the acute treatment for white matter lesion formation episodes, but they are in the spirit of we want to speed up your recovery, not we want to limit the amount of damage. We don't think that we can limit the amount of damage. We think that these are given too late. If we had given them before you'd actually started developing developing symptoms, then maybe they would have reduced the damage. But of course, we don't know that you're actually having an episode of white matter lesion formation until you've actually got the symptoms, by which time it's too late to actually reduce the damage. So we give glucocorticoids, but the spirit with which we do this is to speed up recovery, not to reduce the damage. We wish they reduce the damage, but we don't think they actually do. The damage has been done by the time we're giving them. So, let me just remind you of the mechanism by which glucocorticoids work, just in case you're not familiar with it. So, let's draw a little cell here. So, this can be the cell membrane here, and then here is the nucleus of the cell. Right, so inside all cells within your body, in the cytoplasm, you have a receptor for glucocorticoid molecules, and that receptor is known as the glucocorticoid receptor, which for short I've abbreviated to GR here. So this is the receptor for cortisol, the endogenous glucocorticoid. Now normally this receptor is in the cytoplasm and is not doing anything, and the reason it's trapped in the cytoplasm is it has another protein bound to it, which for some reason I decided to do in a very similar colour. So this is another protein, and this is called heat shock protein 90, HSP90, and I didn't think I'd managed to fit that inside the box, so I've written it along the side. So I'll write out its full name down here. So, the glucocorticoid receptor has bound to it this heat shock protein 90. And heat shock protein 90 is keeping the glucocorticoid receptor trapped in the cytoplasm. And the glucocorticoid receptor, the place it wants to be is inside the nucleus, because inside the nucleus it can actually do stuff. What it's going to do is change gene expression. It's a transcription factor once activated, and that's how it changes the behavior of cells. So, inside loads and loads of cells within your body, you have the glucocorticoid receptor set in the cytoplasm bound to heat shock protein 90. Of course, you'll have loads of glucocorticoid receptors, and they'll all be bound to heat shock protein 90. What then happens is if you have a glucocorticoid molecule, so let's say here, capital G, this box here, this is a glucocorticoid molecule. Glucocorticoid molecules are lipid soluble, so they can cross cell membranes very easily. So they go into the cytoplasm and then they bind to the glucocorticoid receptor, and when they bind, they cause the heat shock protein 90 to detach from the glucocorticoid receptor. So what's going to happen now is here's the glucocorticoid receptor, and here is the glucocorticoid bound to it, and you will notice that the heat shock protein 90 has gone now. So you now have your glucocorticoid receptor glucocorticoid complex. This can now go into the nucleus because it doesn't have that troublesome heat shock protein 90 attached to it anymore that is keeping it chained in the cytoplasm. Inside the nucleus, it then is going to start acting, but in order to act, it's going to have to dimerize with another glucocorticoid receptor that's in the nucleus, and therefore that other glucocorticoid receptor is also going to have a glucocorticoid molecule bound to it. So just drawing this in here, those are the two glucocorticoid receptors, and look, they've both got glucocorticoids attached to them. So this is showing the dimerization, two glucocorticoid receptors bound to each other with glucocorticoid molecules bound to each of them, because the glucocorticoid molecules were the things that allowed them to come into the nucleus in the first place. And this complex of two glucocorticoid receptors with glucocorticoids bound to them, this is now capable of changing gene expression. So I'll put this here. So changes gene expression. It's a transcription factor. It binds to DNA all over the place in the genome and alters the expression of genes. It alters which genes are actually going to be used and have their proteins made, and which genes are not going to have their proteins made. And therefore it changes what proteins you have inside this cell, and therefore it changes the behavior of the cell. So that's the way that glucocorticoids act in the body. So, these molecules are going to have effects all over the body. The effects that we are interested in is the fact that they will go inside cells of the immune system and stop the production of pro-inflammatory mediators. So, for instance, if we go down here, they'll go inside microglial cells and stop the production of pro-inflammatory mediators. They'll go inside of the macrophages and stop the production of pro-inflammatory mediators. They stop the production of pro-inflammatory mediators, and if you stop the production of pro-inflammatory mediators, you stop inflammation, you stop bringing more cells of the immune system into that area. Now, why is this going to help with the healing process? Why is it going to speed up the healing process? Well, remember, in our white matter lesion, by the time you've got the neurological symptoms, all of the myelin sheaths have gone. So demyelination has occurred in the white matter lesion. But we've still got loads of inflammatory cells there. We've still got loads of cells of the immune system there. And we've still got loads of pro-inflammatory mediators there. And the cells of the immune system, they will still be producing these inflammatory mediators, because there will still be some signs of immune complexes present, and those inflammatory mediators that they're still producing, those will still be bringing in more cells of the immune system. In order to actually begin the healing process, we need the inflammatory mediator levels to drop right down. And yes, they will drop down because of the destruction of the myelin sheaths, but that's going to be slower than if we added in glucocorticoids there. So what I'm trying to say here is that if you give glucocorticoids, it's going to lead to the inflammatory mediators in the white matter lesions dropping much quicker than they would do if you didn't give glucocorticoids. So therefore, the removal of inflammatory cells from this area is going to occur much quicker if we give glucocorticoids, because we're not bringing as many immune cells in anymore because of the reduction in the inflammatory mediators that the glucocorticoids have produced. So I just want to explain that again to make sure that I've got my message here. So, in order for the healing process to start beginning, you have to have the destruction of the myelin sheaths. That's happened now. And once the myelin has gone, we've discussed the fact that the cells that are releasing inflammatory mediators, which are the things that are continuing to bring in more cells of the immune system and maintain, therefore, the inflammatory response, those are going to now go down, their levels are going to fall. If we add in a glucocorticoid, their levels will fall much more rapidly than they would if we didn't add in the glucocorticoid. And since the inflammatory mediators falling is the thing that stops more immune cells coming in, that's going to be very, very helpful. So we're going to stop more cells coming in much more rapidly if we add in a glucocorticoid, and therefore the removal of the inflammatory cells from this white matter lesion is going to occur quicker because we haven't got as many immune cells to remove. So overall, the glucocorticoids are going to reduce the release of inflammatory mediators, and therefore the drop in inflammatory mediators is going to occur much more steeply, and therefore we will be able to clear the immune cells from that area quicker because we won't have brought as many in because of those reduced levels of inflammatory mediators. So I hope that's clear how we're hoping that this will actually speed up the recovery by stopping the production of pro-inflammatory mediators from these cells of the immune system. Now, you can only give glucocorticoids short term, so these individuals coming in with an acute episode of white matter lesion formation, they'll be given glucocorticoids for a short period of time and then they'll be stopped. And the reason glucocorticoids should only be given for a short period of time is that they cause all sorts of side effects. Remember, this glucocorticoid receptor is found in loads more cells around the body than just the immune cells, and therefore you're changing the behaviour of cells all over the body. So it leads to a huge number of other effects all over the body that can, if done for a long time, lead to serious side effects indeed. Glucocorticoids over a long period of time lead to something called iatrogenic, meaning caused by doctors, Cushing's syndrome, which is the syndrome that you get when you've got far too much cortisol within your bloodstream. So I'll just put that down as a potential side effect. So in red here, the side effect this can lead to is iatrogenic, meaning that it's caused by doctors, Cushing's, and then I've run out of space, but you could add on syndrome there. Iatrogenic Cushing's makes sense, however. So, what happens in Cushing's syndrome? Well, you tend to gain a huge amount of weight because these things increase your appetite, and all of the weight tends to be put on centrally, so you end up with a very large belly. You also end up with a sort of buffalo hump on your back of fat. You end up with a quite fat, round face known as a moon face. They also thin your skin, they thin your bones, they make you bruise easily, they increase your blood pressure, they increase your risk of developing type 2 diabetes. So taking these drugs for a long time does cause serious problems, so they can only be given for a short period of time, or ideally they should only be given for a short period of time. Okay, so there's our discussion of the acute treatment of multiple sclerosis now finished. Let's go on and discuss the prophylactic treatment of multiple sclerosis. So drugs that we're now going to give to people with multiple sclerosis to prevent them getting recurrent episodes of white matter lesion formation. And I should say, we give the acute treatment to anyone who's had an acute episode of white matter lesion formation. So whether you've just got a clinically isolated syndrome or whether this is your fourth episode and you've got relapsing remitting multiple sclerosis, we can give you the glucocorticoids to try and speed up your recovery. Prophylactic treatment is obviously not going to be given to people who have just had a clinically isolated syndrome. This is going to be reserved for multiple sclerosis recurrent episodes of white matter lesion formation. Specifically, it's only actually used in people with relapsing remitting multiple sclerosis, and the reasons for that are complicated. So you have to understand that in the people with progressive multiple sclerosis, of course, they are accumulating more and more neurological deficits. So these people with progressive multiple sclerosis, they're getting iller and iller. The prophylactic drugs for multiple sclerosis, these stop or aim to stop the formation of new white matter lesions. They are not going to help in the healing of previous white matter lesions, so they are not going to get rid of the symptoms that people with progressive multiple sclerosis already have. In addition, these are very, very strong drugs that come with their own potentially horrible side effects. So in people with progressive multiple sclerosis who are very, very ill, it's thought that they aren't in as good a position to tolerate the side effects of these very strong prophylactic drugs as people with relapsing remitting multiple sclerosis who can actually be extremely healthy. So these drugs are generally only used in relapsing remitting multiple sclerosis rather than in the progressive forms of multiple sclerosis. And I'll stress again, they will not help in healing the previously formed white matter lesions. They aim at preventing the formation of new white matter lesions, not in healing previously formed white matter lesions. Okay, so let's discuss these prophylactic treatments for multiple sclerosis then. So I have, how many do I have to tell you about? I think I have 7 of these to talk about, so let's begin. So the first one that I'm going to mention is something called interferon beta, which for short is abbreviated down to IFN beta. So this stands for interferon beta. Now, what is this? This is a protein that is part of the human proteome. It's a protein that our body can produce and which clever biomedical engineers have found a way to actually produce outside of the body. So using recombinant DNA technology, we have found a way to actually produce this protein, interferon beta. And giving this protein to people is actually somewhat effective at preventing relapses in the multiple sclerosis, in preventing new episodes of white matter lesion formation. So the question is, how is it actually effective? Now, I should say that a lot of these drugs that are given prophylactically for multiple sclerosis, we don't actually have a brilliant understanding of how they actually work. So a lot of what I'm going to say is extremely controversial, and it's almost just potential possibilities for how it could work, a logical explanation for how it could work, but it's all theory. It's potentially very wrong. The official line for most of these drugs is we do not know how it works. All I can offer you is a potential logical explanation as to how this drug may work to prevent future white matter lesion development. So that's the spirit with which we're doing this. We don't really know how most of these drugs work. So interferon beta, let me tell you about what the normal function of interferon beta is, and then I can try to potentially justify why this might stop future episodes of white matter lesion formation. So interferon beta is an example of what we call a type 1 interferon. So there are other type 1 interferons as well, and they all do the same kind of thing, and they're involved in fighting viruses. They're involved in inducing what we call an antiviral state in cells. So let me sort of motivate this by actually giving you the scenario. So let's say we have some cells here. So here is one human cell, here's another human cell, and I'll draw one more human cell. Okay, and let's say that we have a virus. So in red here, this is going to represent the virus. Let's say this virus enters this human cell here and starts using its machinery to make loads more of these copies of the virus, i.e. the virus is using the cell to replicate. What this cell can now do, the virally infected cell, it can now start releasing type 1 interferon molecules, including interferon beta. And what these molecules go and do is they go and work on the surrounding cells that have potentially not been infected by the virus yet. And they bind to receptors on the surface of the surrounding cells, and they tell those cells there's a virus about, you need to prepare. And they make the cells go into an antiviral state. And what is meant by an antiviral state is a state where the virus cannot easily actually use that cell for its own purposes. In particular, the way this actually works is the cell will shut down protein synthesis machinery, so it will stop its production of proteins. And if it shuts down the protein synthesis machinery, then the virus isn't going to be able to use that protein synthesis machinery to make its own proteins, and hence the cell is very difficult for this virus to actually hijack and abuse. So, let me summarize what I've just told you again. So if we've got a virus here, and it's infecting one of the human cells here, this cell can recognize the fact that it is now infected by a virus because its behavior changes hugely, and it can start producing molecules called type 1 interferons, of which interferon beta is an example. So it can start releasing interferon beta and other type 1 interferon molecules. These diffuse off to the surrounding cells, bind to receptors on their surface, and cause those surrounding cells to go into an antiviral state where they turn protein synthesis effectively completely off. They shut down protein synthesis. Then, if a virus does come along and try and infect this cell, if it's in the antiviral state, the virus cannot use that cell to replicate itself because the protein synthesis machinery is all shut down, so it can't make any of its own proteins. And this is a major defensive mechanism used to prevent viral infections. So, we give this protein, interferon beta, to people with relapsing remitting multiple sclerosis to try and prevent them getting future episodes of white matter lesion formation. So how on earth is this going to stop people getting future episodes of white matter lesion formation? Well, here's a possible explanation. This is not the official explanation, this is a possible explanation. The official explanation is we don't know. A possible logical way that this could occur is maybe if you give high doses of interferon beta to people, it puts their cells into a chronic antiviral state, and therefore they are less susceptible to getting severe viral infections. Maybe this protects people from getting flu, which is caused by the influenza virus. It will protect you maybe against getting colds, which are caused by rhinoviruses and coronaviruses. It might protect you against getting gastroenteritis, which is often caused by norovirus or rotavirus. So maybe it actually protects you from getting severe infections, which then would cause elevated levels of pro-inflammatory mediators, interleukin-1 and tumor necrosis factor alpha, which then cause the episodes of blood-brain barrier penetration. So that might be a logical reason as to why giving high doses of interferon beta, recombinant interferon beta, to people with relapsing remitting multiple sclerosis might reduce the number of episodes of white matter lesion formation that they actually suffer from. So summarising what I have told you, interferon beta is a human protein. Biomedical engineers have managed to produce this protein, a recombinant version of this protein, and we can give it to people with relapsing remitting multiple sclerosis. It will go into their bloodstream and somehow it prevents them getting future white matter lesion formation. Now, of course, it doesn't completely stop it. Some people will be given this drug and maybe won't ever get another episode of white matter lesion formation in their life. Fantastic. Many people will be given this drug and will still get episodes of white matter lesion formation, maybe less frequently than they would have if they hadn't been given the drug. So how does this possibly work? Well, interferon beta is a type 1 interferon. It induces an antiviral state in cells, so it is possible that by giving high-dose interferon beta, we put people in a chronically protected state against viral infections such as flu and gastroenteritis, and therefore we are preventing episodes of blood-brain barrier penetration, because when you get severe infections, those are the things that prompt an episode of blood-brain barrier penetration. So that's a plausible explanation as to how interferon beta might be effective at treating relapsing remitting multiple sclerosis. Okay, next. So the next drugs that we're going to discuss, I'm going to put in a category called immunomodulators. And this isn't just me that puts it in this category, this is a category that many people use. So there's 4 drugs that are going to be in this category of immunomodulators. So the first question is, what actually is an immunomodulator? An immunomodulator is a drug that is going to change the function of the adaptive immune system. It's going to change the way the adaptive immune system works. It's going to modulate the adaptive immune system. It is not the same as an immunosuppressant. I'm going to discuss 4 drugs that are in this category of immunomodulators, and then we'll discuss 2 more drugs that are in the category of immunosuppressant. An immunosuppressant is a scarier drug. An immunosuppressant is something that turns off the adaptive immune response. Immunomodulators, they don't do that. They're not quite things that turn off the adaptive immune response. They interfere with the adaptive immune response, but they don't quite turn it off in the way that immunosuppressants do. So they're less scary drugs than the immunosuppressants. So let's talk about examples then of immunomodulators, and I'm afraid in this category of drugs, this is the category that I'm going to say most we don't really know how it works. You'll see me saying we don't really know how this drug works for quite a few of these, and I can't really offer you any more than the fact that it interferes with the adaptive immune response and the maintenance of the autoreactive adaptive immune response. So let's get a new colour to do this in. So we'll go to a rich blue colour. So the first drug that I want to discuss is a drug called glatiramer acetate. So glatiramer acetate. Now, this drug has a brand name that is very, very famous, so I'm going to actually give you the brand name. This is the drug that most people will refer to as Copaxone because it's a simpler name than glatiramer acetate. So Copaxone is not the actual official name for this, it's the brand name. The official name is glatiramer acetate. I think this is actually a little bit of a too dark colour, so we'll go now to aquamarine. So, this drug glatiramer acetate, firstly let me just tell you a little bit about it. It is actually a polypeptide, and it's a polypeptide that we produce, again, clever biomedical engineers produce, out of 4 amino acids. So let me tell you about what those 4 amino acids are. So the 4 amino acids are the amino acid glutamate, the amino acid alanine, the amino acid lysine, and I'm using the 3-letter abbreviations for these amino acids, and the amino acid tyrosine. So these are 4 amino acids, and we can make great big polypeptides using these amino acids. And that's what glatiramer acetate is. I'm just going to actually show you the structure of these amino acids just to remind you, so that you've got a better idea of what glatiramer acetate is actually consisting of. So, let me firstly remind you of the basic structure of an amino acid within a polypeptide. So, if this line here represents a polypeptide, let's take a single amino acid out of that polypeptide and draw it over here. So, the basic core structure of an amino acid is that you have the amino group at one end, which will be bound then to the carboxylic acid group of the amino acid in front of it, and this is considered the front side of the amino acid, and you'll have the amino acid in front of it bound here, so the amino acid up here, and it will be bound by an amide link, which is this bond here, to the carboxylic acid group of the amino acid in front. So there's the amine group. Then you have the alpha carbon here with a hydrogen coming off it, and also the important R group, which is the group that is different between the different amino acids. After that, you have the carboxylic acid group of the amino acid, which will be bound to the amino group of the next amino acid along, again by an amide link. So this is the amino acid residue structure within a polypeptide. And in the different amino acids that are used in polypeptides in humans, you'll have different R groups in this position. So let me just tell you about the R groups, or remind you of the R groups of these 4 amino acids here. So, starting off with glutamate. Glutamate has a carboxylic acid R group consisting of 3 carbons in length. So it's this R group. So there's the carboxylic acid group, and then off these 2 carbons here you have hydrogen atoms coming off, and then this will be attached to the alpha carbon. So put this in place of the R group. That is the R group of glutamate. So that's glutamate. Alanine is an extremely simple amino acid. Its R group is literally just a methyl group, so it has a carbon with 3 hydrogens coming off in place of that R group. Lysine is a little bit more complicated. It's 4 carbons in length with an amino group at the end, so 1, 2, 3, 4, and then right at the end you have an amino group, NH2 here, and then all of the carbons along here, those are saturated with hydrogen atoms coming off like so. And then finally, the 4th amino acid, tyrosine here, its R group I will draw up here, and it has a methylene group, so a carbon with 2 hydrogens coming off it, like so. And then it has a benzene ring, which I'll just draw in the classical way to show a benzene ring, as a hexagon with a circle in the middle, and then it has an alcohol group coming off that benzene ring. So that's the R group of tyrosine. So, those are the structures of these 4 amino acids. We can build polypeptides, a polypeptide like so, out of these 4 amino acids, where in each one of the positions we will have one of these 4 amino acids. Glatiramer acetate is a subcutaneous injection, so that's the way that it's administered. So I'll just put that here. It is a subcutaneous injection, so S/C means that it's given underneath the skin, subcutaneous, and it consists of loads of polypeptides that consist of these 4 amino acids. Now, how does this work then? It is believed that somehow giving these polypeptides of these 4 amino acids actually prevents the continual maintenance of the autoreactive adaptive immune response. So remember, in order for multiple sclerosis to go on and on and on for years and years and years, you have to maintain that autoreactive adaptive immune response. You have to maintain your population of autoreactive effector CD4 positive T cells, and you have to maintain your population of autoreactive antibodies within the bloodstream. So you have to maintain your population of the autoreactive effector B cells in the bone marrow that are producing those antibodies. For that to occur, you're having to continually reactivate the adaptive immune system because the effector cells do not live that long. They live for a few weeks to months. After that, they are removed from the body. So you have to continually reactivate the memory CD4 positive T cells and the memory B cells in order to maintain that. Glatiramer acetate is believed to somehow get in the way of the continual renewal of the adaptive immune response. It's believed to interfere with the maintenance of the autoreactive adaptive immune response. How does it do this? We don't know. It is believed that potentially within these polypeptides that you're going to have within the drug, that if you get the right combination of glutamate, alanine, lysine, and tyrosine, that might contain the peptide fragment of the CNS myelin protein. So I'll say that again. So remember back the CD4 positive T cells that are directed against the CNS myelin protein, they're directed against a specific little peptide fragment of it. We believe that that peptide fragment might be made up just of glutamate, alanine, lysine, and tyrosine amino acids. So if we make enough polymers out of these amino acids, somewhere it's likely that in one of those polypeptides is that same peptide fragment that is actually present within the CNS myelin protein. So the idea is that with glatiramer acetate, we might be presenting the body with loads of this peptide fragment that it has developed an autoreactive CD4 positive T cell clone directed against. And somehow doing that seems to inhibit the continual maintenance of the activation of that CD4 positive T cell clone. But I'm afraid that's the sum of what I can tell you about how glatiramer acetate works. We don't know. Okay, there is research going on to try and understand the significance of this drug, because indeed you would think that by studying this drug, given that we think it may well contain the peptide fragment that the autoreactive CD4 positive T cell clone is directed against, that it would give rise to a lot of answers potentially about what protein we're actually attacking in multiple sclerosis. So there's a lot of hope that by studying this drug we will learn some interesting truths about this disease. But at present, that's the best I can do as far as explaining how this drug works. So to summarize, glatiramer acetate contains loads of polypeptides made of the amino acids glutamate, alanine, lysine, and tyrosine, and somehow giving this subcutaneously stops the maintenance of the autoreactive adaptive immune response, and therefore is effective at stopping further white matter lesion formation in multiple sclerosis, is the hope. Because of course, if you stop the autoreactive adaptive immune response, then the levels of effector CD4 positive T cells and antibodies within the bloodstream will go down, and if those go down, you've taken out step 2 of the pathogenesis, and therefore you're going to stop the formation of future white matter lesions. So we'll have a break here. In the next video we will talk about 3 more immunomodulators and then 2 immunosuppressants finally.